Hi everyone, Assalamu alaikum. Today's topic is epitope. First, like and subscribe my YouTube channel. So let's start our topic. An epitope, also known as antigenic determinant, is the part of an antigen that is recognized by the immune system, specifically by antibodies, B cells or T cells. The part of an antibody that binds to the epitope is called a paratope. Although epitopes are usually non-self proteins, sequences derived from the host that can be recognized, as in the case of autoimmune disease, are also epitopes. The epitopes of protein and antigens are divided into two categories: conformational epitopes and linear epitopes. These are based on their structure and interaction with the paratope. Conformational and linear epitopes interact with the paratope based on the 3D conformation adopted by the epitope, which is determined by the surface features of the involved epitope residues and the shape or the tertiary structure of other segments of antigen. A conformational epitope is formed by the 3D conformation adopted by the interaction of discontiguous amino acid residues. In contrast, a linear epitope is formed by the 3D conformation adopted by the interaction of contiguous amino acid residues. A linear epitope is not determined solely by the primary structure of involved amino acid. Residues that flank such amino acid residues, as well as more distant amino acid residues of the antigen, affect the ability of primary structure residues to adopt the epitope 3D conformation. 90% of epitopes are conformation. Next function. T cell epitopes. T cell epitopes are presented on the surface of an antigen presenting cell, where they are bound to major histocompatibility complex (MHC) molecules. In humans, professional antigen presenting cells are specialized to present MHC class II peptides, whereas most nucleated somatic cells present MHC class I peptides. T cell epitopes presented by MHC class I molecules are typically peptides between 8 and 11 amino acids in length, whereas MHC class II molecules present longer peptides, 13 to 17 amino acids in length. And non-classical MHC molecules also present non-peptidic epitopes such as glycolipids. Next, B cell epitopes. The part of the antigen that immunoglobulin or antibodies bind to is called a B cell epitope. B cell epitopes can be divided into two groups: conformational or linear. B cell epitopes are mainly conformational. There are additional epitope types when the quaternary structure is considered. Epitopes that are masked when protein subunits aggregate are called cryptotopes. Neotopes are epitopes that are only recognized while in a specific quaternary structure, and the residues of the epitope can span multiple protein subunits. Neotopes are not recognized once the subunits dissociate. Next, cross activity. Epitopes are sometimes cross reactive. This property is exploited by the immune system in regulation by anti-idiotypic antibodies, originally proposed by Nobel laureate Niels Katz. If an antibody binds to an antigen's epitope, The paratope could become the epitope for another antibody that will then bind to the epitope for another that bind to it. If the second antibody is of IgM class, its binding can upregulate the immune response. If the second antibody is of IgG class, its binding can downregulate the immune response. So it was all about epitope and its function. Thank you.